This is Emily Muff, and we're going to discuss my research project proposal. So the start of this project stemmed from my interest in human gut health and all the fascinating research that's been coming out of that. There's been connections made from the microbial count in the human gut to GI diseases and conditions, cancer, and mental health. So while a relatively new treatment, fecal transplants have been the answer to those suffering with issues stemming from a low microbial count such as C. diff. So this same idea can somewhat be carried over to the plant world. So a plant is going to thrive in a soil that has a higher microbial count than one living in a nutrient-poor soil. So with that in mind, I chose to research soil microbial diversity, specifically two of the main soil inoculates on the market right now, which is a rhizobium and a mycorrhizal fungi. So somewhat comparable to fecal transplants, these soil inoculants are available to large-scale agricultural companies as well as the at-home gardener to further enrich their soil. So fungi specifically decompose, organic matter, promote soil aggregation, play a role in nutrient cycling, and biocontrol of plant pathogens. So through evolution and natural selection, fungi are found in almost every environment in which temperature and pH vary greatly. Through the release of extracellular enzymes, these fungi are able to break down organic compounds from plant and human components. While soil aggregation, usually is associated, with organisms such as earthworms, fungi can contribute to this essential mechanical process through the growth and extension of their hyphae and mycelium. Bacteria make up an astonishing amount of soil biomass. Their weight can be comparable to 1 or 2 cows. Specific bacteria that inhabit the root soil interfaces, known as the rhizosphere, break down organic matter by means of extracellular digestion via enzymes, similar to the way fungi do. Bacteria within the soil is also essential for nitrogen and phosphorus cycling, making these nutrients available for plant uptake. Separately, bacteria and fungi play instrumental roles in the soil and their contribution to plant health, though it's suggested that the interactions between microbial components themselves is just as influential. It's been shown that specific bacterial populations within the rhizosphere increased with fungal colonized plants. So additionally, it's been shown that plant associated with specific fungi and bacteria, which I'll discuss here shortly, produced increased levels of phytohormones. Mycorrhizal fungi are arguably the most important class of fungi. So they form mutualistic relationships with roots of the plants, and through this relationship, the surface area of the plant's roots is increased, which thus increases the intake of nutrients. They can also assist in drought resistance, again, through the extension of the plant roots via the fungi, allowing plant to take advantage of the available resources within the soil that it otherwise could not. Additionally, because of the low mobility of phosphate ions, the reaching hyphae and mycelium can acquire these ions outside the phosphate-depleted soil that surrounds the plant roots. And bacteria belonging to the genus Rhizobium form symbiotic relationships with the plant roots via little root nodules. These root-colonizing bacteria are referred to as plant growth-promoting rhizobacteria, PGPR for short, and are involved in nutrient cycling and protect the plant from potential pathogens. So a lot of us know that nitrogen is an essential nutrient for plant health and development, though it's not usable in its atmospheric form. So these nodule-living bacteria convert atmospheric nitrogen into usable nitrogen. And while this relationship has been prevalent in legumes, it has been shown that specific bradyrhizobia have the potential to act in similar ways to plant growth-promoting rhizobium on non-legumes, specifically the humble radish. Each microbe within the soil has a role to play, and together these roles result in a healthier and more abundant plant. High microbial diversity in soil has been shown to positively affect carbon decomposition, be essential in nutrient cycling, and protect plants from pathogens. While mycorrhizal fungi and rhizobium are naturally occurring in the soil, not everyone has access to such nutrient-rich soil. Through the use of inoculants, however, that soil can produce healthy plants. With increasing population means decreasing agricultural lands. It's been shown that crop yield significantly has increased through the use of soil inoculants. This can help large-scale farmers as well as the at-home gardener to maximize their harvest without the use of pesticides. So I predict that the soil with the highest microbial diversity will produce the healthiest crop. So I built an 8-foot by 3-foot raised garden bed that's going to have 5 separate compartments. And each compartment is going to have polyethylene sheets to ensure a watertight seal between the compartments, so I know no leaching is going to be happening here. Each compartment is going to represent a different level of soil microbial diversity. I obtained some Bridgeport silt loam soil from University of Rhode Island, and to start out, I'm going to lay it out on a tarp and mix it to ensure an even dispersal of all the soil components. And then an equal amount of soil is going to be placed in each compartment with the following preparations. So the soil in compartment 1, 2, 3, and 4 are going to be sterilized per the following procedure, while compartment 5 is going to contain untreated soil. So, per reference of a previous study done and standard autoclave operating procedure, 3 800 milliliter glass jars are going to be filled with approximately 4 inches of soil and placed in an 8-quart Instant Pot with half a liter of deionized water added to the bottom of the pressure cooker to generate steam. And then I'm going to let the cooker, it's going to be set to a slow cook setting with high temperature and high pressure for 60 minutes. This is in accordance with that same study, which found that 60 minutes was the required time to kill both bacterial and fungal microorganisms in soil. Maintaining an aseptic technique, the sterilized soil will then be transferred to 5-gallon rigid plastic construction buckets with an airtight lid. Identical cycles are going to be completed until an adequate soil amount is sterilized. So, compartment 1 is going to contain only sterilized soil. Compartment 2 is going to contain the sterilized soil with the PGPR inoculant purchased from seedworldusa.com, and the inoculant will be applied per the manufacturer's instruction at a ratio of 6 ounces of inoculant to 50 pounds of seeds. Compartment 3 is going to have sterilized soil and the mycorrhizal inoculant purchased from arbicoorganics.com. The inoculant will also be applied per the manufacturer's instruction, which states to mix evenly between 1/4 and 3/4 pounds of the inoculant per cubic yard of soil. Compartment 4 is going to have the sterilized soil and both inoculants that will be applied per the methods previously stated. And then 2 radish seeds are going to be directly sown in at a 1/2 inch depth at 1 inch apart, and they will be thinned down to 1 plant when they reach 1 inch tall. So I'll have 20 plants in each compartment. Weekly measurements of plant height, leaf width, and ambient temperature will be documented until harvest. And qualitative measures such as leaf and stem vigor, leaf color, and overall appearance will be documented weekly. Harvested roots will be measured in length, width, mass, and leaf-to-root ratio. Qualitative measures such as overall flesh appearance will also be documented. So, because of my limitations here at home, I'm unable to test for what is actually living within the Bridgeport soil. There's a chance that the microbial diversity is already low, but I'm going to assume that it is not. Also, while there has been research conducted on the use of an Instant Pot in place of an autoclave, I cannot test whether my Instant Pot will produce similar results. Lastly, after sterilization, I cannot determine the true sterility of the soil, nor can I maintain a true sterile environment without the use of proper air circulation. So some level of contamination is to be expected. Like I stated earlier, with population increasing and land decreasing, we need as much help as we can get in order to increase our crop yields. GMO seeds and inoculants could be a part of that answer. While I'm not testing different kinds of seeds, such as GMO, non-GMO, or heirloom, something like that, perhaps paired with organic versus non-organic growing methods, would be a suggestion for further research.